Okay, this video here is to help walk you through Notes 3 on Intro to Genetics and Punnett squares. Now, if you're confused or you don't want to watch a video to walk you through it, you can attend the review session on Wednesday, Thursday, or Friday for your period and do it in real time. But if you prefer to do it on your own, you can watch this video and that'll help. So part 1 is all about the history of how did we figure out this whole genetics thing. So genetics is really the study of inheritance and how we get the traits that we do and why we look the way we do. And the father of genetics is this guy, Gregor Mendel, who lived in the mid 1800s, and he worked in a monastery, which is where monks work. That's why he's wearing that monk outfit that you can kind of see right here in the picture. He worked in a monastery and he studied pea plants. So like in the summer, if you've ever grown sugar snap peas, those are examples of the plants he studied. He was really careful in his experiment design, and pea plants were really good to use because it was easy to breed the plant that he wanted to. So before Mendel did all of his experiments, everyone thought that that we inherited traits through a blended hypothesis or blended inheritance. And so click on this simulation link and pause this video. And in the simulation, you're going to play around with thinking about what a blended hypothesis would mean. And then you're going to take a screenshot of this completed chart after you have dragged some of these into the various blanks. So we'll do one together to kind of think about it. So think about a blended hypothesis. It's a blend. And so if we took parents, these are representing the parents over here. If we took one parent with wrinkled pea seeds and one with round pea seeds, well, it might make sense that they would have round, wrinkled babies. So the blended hypothesis is that you are a blend of your parents. So if you are medium height, it's because you have one tall parent and one short parent. And that's in general what people thought. You have brown hair because you have one black-haired parent and one blonde-haired parent. But Mendel's experiments disproved this, and we can see that in the actual outcome over here. Because when wrinkled and round parents came together, they had all round babies. And when a parent with white flowers and a parent with purple flowers came together, they had all purple babies. Same with height. A short and a tall parent had all tall babies. And so clearly Mendel's results disproved the theory of blended inheritance, and instead they proved a theory of dominance. Okay, so the dominant allele or dominant phenotype, physical trait, is what was shown up. So for seed shape, what was dominant? Well, round, because that's what showed up. So what was then recessive? Well, recessive was wrinkled. So if you haven't already, pause the video to do this section on 1, 2, and 3. You'll notice on question 1, you're taking a screenshot and inserting it. The screenshot is like the chart that I was just drawing on over here, except it's filled in with the blended hypothesis. To take a screenshot, you go Command-Shift-4 and it will save the image onto your desktop. Then you can just drag that image into your notes. Okay, and the video I'm going to keep going. So what we're going to do in this next section, Mendel's crosses, we are going to actually dive into Punnett square and the details of what was happening with Mendel's tall and short plants. So we're going to consider height. So in order to edit this, you need to double click on it on your Google Doc. So double click and then let's go in there. So the parent generation P generation. P1 cross is talking about the parents. We're considering one tall and one short parent here. Now we're going to use the letter T, but we could choose to use any letter. So above in the simulation, we saw that tall was dominant. Okay, so capital T is going to be dominant. Short was recessive. And if you don't remember, lowercase t, what these words mean from our human traits lab earlier in the week, you are going to define them again in the next section, so you'll get another chance. So let's take a look at these crosses. We are going to assume the parents are homozygous, meaning the same allele. So remember, you have two alleles, or plants have two alleles per gene, so two letters. So the parent that's tall, we're assuming is homozygous, big T, big T. The parent that's short, little t, little t. Okay, homozygous recessive. So now, meiosis would happen here to separate alleles into sperm and egg. So if we look, we know that meiosis would happen and only one allele goes through meiosis. Or another way to think of it is that sperm and egg are haploid, meaning they have half of the chromosomes or half of the letters that the parent cells have. Little t, little t. Okay, now our last row is showing all of the possible combinations for fertilized offspring, which is quite easy here because if we look at the blue male plant, the only option is that that male plant is going to give a big T because it only has a big T to give. The female plant is going to give a little t because it only has a little t to give. Now that image with all those arrows and confusing circles, instead of showing like that, we draw Punnett squares. So let's practice doing a Punnett square instead to show what the options are for the babies, fertilized offspring babies, from these two parents. So when we're doing a Punnett square, the first step is that we have to figure out what the parent genotypes are. Remember the parent letters, and we did. It's big T, and little t, little t. Once we figure that out, we put the parents on the top and on the side of a Punnett square. It does not matter which you put where. You could put the big T's on the left and the little t's up top. It wouldn't matter. Now what we do is we go through and we put the letters in the other boxes. We always do capital big letters first. So what I'm going to do is I'm going to do big T first, big T first, big T first, big T first. Then I'm going to go through and go little t, little t, little t, oops, little t. Now whenever we do Punnett squares, we also do ratios or percents. Whenever it says ratio, you can always do a percent too of the genotypes and the phenotypes. So let's do that too. Remember, genotype is letter combination. So the genotype is big T, little t, and it's 100%. Or the ratio is just 1. Okay? Now, phenotype ratios also 100%. Or the ratio would just be 1. And instead of writing the letter combination, we write the physical trait. All of the babies are going to be tall, and that's what Mendel saw. Now, we can see that in the picture here. In this second image, in this second cross or breed, a cross is like saying you're just going to breed two plants or animals or whatever. We're going to take these plants, so the babies that are these or these ones, and we're going to breed them. So notice they are heterozygous. So let's go down. Our parents here are heterozygous. So big T, little t, big T, little t. So then meiosis would happen here to separate alleles into sperm and egg. Big T, little t, big T, little t. Okay, now we're going to do a Punnett square with those parents. So again, we put big T's first, little t's second, big T's first, little t's second. Then when we go through, still do big T's first, big T's first, little t second, little t second, little t. Our ratios here are a little trickier, or our percents. Remember, you can think about them either way. So let's do this a little more carefully. So for the genotypes, we want the letter combinations. So notice there's one genotype that is big T, and we could also say that one of those squares is 25%. Okay, so 25% is big T, or 1. Now, ratio, or I usually kind of do it with dots. So then the next part is going to be about big T, little t, and there's 2 there. So the ratio is 2, big T, little t, or we could say 50% big T, little t. Okay, then we have a ratio again of 1 because little t is only 1 square and it's 25%. Right there. And then the big T's are separate too. So when you're doing ratios or percents, make sure you don't just say 50% or just 2. You have to say 50% big T, little t. So make sure that those go together. Okay, now if we think about the phenotypes, are any of these the same? Well, yes, let's do it for tall. How many of these are tall? This one is tall, this one is tall, and this one is tall because tall is dominant. So we have 3 tall, or 75%. Okay, so 3 is the ratio. So the ratio is 3 to 1 short, or 25% short. Now, you can do ratios or percents. When I'm making keys for things, I'll do both, but I find that most students prefer to use percents, but you can use whichever makes sense to you. So what are the steps to doing a Punnett square? Well, what did we do? Step 1 was determine parent genotypes. Okay. Step 2 was then place parent alleles on sides. Okay, that's when we would say put those there and those there. And then our last step is cross letters or alleles, or essentially fill in the Punnett square. And then we always do percents or ratios, something to analyze the information. So that is the end of the part of these notes that I'm going to guide you through, because you can try the rest on your own. Part 2, there's a very short video. Okay, and you are defining 6 of the most important vocabulary words. There's images there to help you too. The video is like 3 minutes, so it won't take too long, but pause it as you need to. Watch the video again. You'll see my recommendation is to just watch the video once, take no notes, just watch, then watch it again. The repetition you get in the classroom, but you don't get it when you're doing it on your own at home. So it makes it harder to learn when you don't get as much repetition. Part 3, you have another video here from the Amoeba Sisters, and it goes along with vocabulary through an example of guinea pigs. And there's weird hairless naked guinea pigs, and then there's hairy guinea pigs. So those are the phenotypes, and you kind of are going through. There's a few examples and questions asking you about that case study of guinea pigs. At the end, you'll notice here it says want more practice. This is optional. There is a link to a Google Doc with more practice and then a key as well. Even if you're just a little bit confused though, I would recommend doing it. At least look at the key and see if it makes sense and kind of skim through it. You are not getting nearly as much Punnett Square practice through virtual school as we would be doing in the classroom. So you need to kind of try to think to yourself, do I understand these Punnett Squares? Do I need more practice? Where's my level of mastery right now? So that's the end of this video. Email me questions, join a conference. I'm here to help.